Right now, as you're watching this, billions of cells inside your body are making a life or death decision. It's a process called apoptosis, or programmed cell death. You could almost call it the art of cellular suicide, and honestly, it's one of the most important things happening inside you.
So let's just get right to it with the big question. Do our cells actually commit suicide? And the answer is a huge, resounding yes. It's happening constantly, all the time, and believe it or not, you literally wouldn't be alive without it.
Now, to really get this, we have to draw a clear line between two very different ways a cell can die. First, you've got apoptosis. Think of this as a planned, quiet suicide. The cell is incredibly tidy about it. It packs up its own things, dismantles itself, and just disappears. No fuss. But then you have necrosis. This is the complete opposite. This is like a traumatic murder. It's chaotic, it's messy, the cell literally bursts open, spills its guts everywhere, causing a big inflammatory scene for all the neighboring cells.
Okay, this all leads to a really fundamental question, right? Why would our bodies want their own cells to die? I mean, it seems totally counterintuitive, but this process, this cellular suicide, is absolutely crucial for keeping you healthy.
It really boils down to two main reasons. First, a cell might just not be needed anymore. Its job is done, it's served its purpose, and it's time to get out of the way to make room for new cells. But the second reason is much more serious. This is when a cell's DNA gets so badly damaged that it just can't be fixed. And to stop that cell from going rogue and becoming something dangerous, like a cancer cell, the body basically orders it to take itself out.
So how does a cell actually carry out this self-destruct order? Well, it has a built-in demolition crew, a team of specialized enzymes called caspases. You can think of these guys as the executioners. They're just lying dormant inside the cell, totally inactive, just waiting for the signal. But the moment they get that green light, they spring into action and start systematically dismantling the cell from the inside out.
And that signal, the one that wakes up the caspases, can come from two completely different places. This leads to two distinct pathways. The best way to think about it is like two different stories, but no matter how they begin, they both have the exact same ending, a clean, quiet cell death.
So, our first story starts with a phone call from the outside. This is what we call the extrinsic pathway. This is where another cell, usually a part of your immune system, shows up and personally delivers the death sentence.
So here's how that works. A signaling molecule comes along and latches onto a special death receptor on the cell's surface, kind of like a key fitting into a lock. Once enough of these keys are in their locks, the receptors all cluster together, and that forms a sort of docking station inside the cell. That station's job? To wake up the first set of caspases, the initiators. And that's it. The countdown to demolition has officially begun.
All right, let's get to our second story. This one doesn't start with a call from the outside. It starts with an alarm bell ringing from inside the cell. This is the intrinsic pathway, and it's usually set off by some kind of severe internal damage. This whole pathway is really a story about a very delicate balance. See, the cell's powerhouses, the mitochondria, are guarded by two opposing groups of proteins. You have the gatekeepers, which are anti-apoptotic. Their job is to prevent cell death. And then you have the gate openers, which are pro-apoptotic, and they're ready to kick things off. As long as the cell is healthy, the gatekeepers are in charge.
But what happens when the cell's DNA is damaged beyond repair? Well, that balance of power shifts. The gate opener proteins take over. They overpower the gatekeepers and they pry open the gates on the mitochondria. This causes a really critical molecule, cytochrome C, to start leaking out into the rest of the cell. And that leak, that is the point of no return. The escaped cytochrome C then goes and activates a different initiator caspase, and just like that, the internal alarm has been sounded.
So, there you have it. Two totally different stories, right? One starts with an outside call, the other with an inside alarm. But it doesn't matter how it started. Both of these paths now merge for one final dramatic act. And this right here is the critical moment. Those initiator caspases we talked about, from both pathways, they really only have one job: to find and activate the final executioner caspases. These are the real demolition crew, and once they're switched on, there's no going back. The cell's fate is completely sealed. Okay, so what does this demolition actually look like? It is not an explosion. It is a controlled, systematic, and frankly, beautiful breakdown. This is where we really see the difference between that quiet suicide and the messy murder we mentioned.
First, the cell starts to shrink, kind of pulling away from its neighbors. Its nucleus gets all condensed and breaks into fragments. Then the cell membrane starts to do this weird bubbling thing. It's called blebbing. These little bubbles, or blebs, then pinch off into these perfectly neat little packages called apoptotic bodies. Each one contains a piece of the dismantled cell. And finally, immune cells show up to do the final cleanup, just gobbling up these packages so that not a single trace is left behind. No mess and absolutely no inflammation.
You know, this whole process is so quiet and efficient that it can be tricky to even spot it happening. So how do scientists and doctors play detective? How do they act like forensic investigators to figure out if a cell is undergoing apoptosis?
Well, they have a couple of really clever tricks. One involves using a special marker that looks for a molecular eat-me signal. See, during apoptosis, a molecule that's normally on the inside of the cell membrane flips to the outside, basically waving a flag for the cleanup crew. This marker only binds to that flipped molecule, making dying cells light up. The other method looks at the DNA. Remember those executioner caspases? Well, they chop up the DNA into very specific, predictable lengths. And when you separate those DNA fragments, they form this unique stepladder pattern. It's a dead giveaway for apoptosis.
So let's bring this all home. The single most important thing to take away from all of this is that fundamental difference, that core analogy, the cell's orderly suicide versus its traumatic murder. The contrast really couldn't be more stark. Apoptosis is a surgical strike. A single cell shrinks down, packages itself up, and just disappears without causing any trouble. Necrosis, on the other hand, is collateral damage. A whole group of cells swells up and bursts, spilling their contents and creating a chaotic, inflammatory crime scene. One is absolutely essential for your health, the other is a sign that something has gone very wrong. So apoptosis is this incredible masterpiece of biological engineering. It's essential for shaping our bodies, for getting rid of old cells, and for protecting us from disease. But that leaves us with one last, really crucial question. If our bodies are such masters of this clean demolition, what happens when the process goes wrong? What happens when cells that should die don't? Or when cells that are perfectly healthy get the signal to die by mistake? Well, that's a whole other story.